translation, one of the basic things a cell does all the time. And for transcription and translation, basically, we're talking about making RNA and making proteins. So first, let me tell you about the different types of RNA. Even though you only know about a couple of them, there really are 11 different types of RNA. And we're going to talk about 3 of them in this class, although one of them you probably didn't even know it had RNA in it. So the first type is messenger RNA. And messenger RNA is going to be abbreviated as a small m in front of RNA. And it's messenger RNA that's going to be coding for the protein. So you can see we need to make the messenger RNA before we can make the protein. The second type of RNA is called ribosomal RNA. That's inside your ribosomes. So we're just going to be talking about ribosomes. We really are not going to be talking about the ribosomal RNA at all. But as you know, ribosomes are the location in which proteins are made. So it does come into play. That's where the messenger RNA is going to have to visit in order to make the proteins. And the third type of RNA is called transfer RNA. And so it's abbreviated with small t in front of the RNA. And it is unique in that it has 2 binding sites. On one end, it's going to bind to the amino acid, and the other end is going to bind to the RNA so that it's going to translate the messenger RNA into proteins. So what is transcription and what is translation? Well, transcription is just transcribing something. We are talking the same language, and the language for transcription is nucleic acid language. Whereas translation, we are going from one language to another language, just like when you translate between Spanish and English. And so the 2 languages we're talking about translating between are between nucleic acid language and the language of proteins. So in transcription, this is going to happen within the nucleus of this cell. And we are going to be using a DNA template to form what we're calling a pre-messenger RNA. And then that pre-messenger RNA gets a little bit of processing, a little nip-tuck, and then it is our messenger RNA, and it can leave the nucleus through the nuclear pores and to enter the cytosol. And in the cytosol is where the ribosomes are located, and the messenger RNA will attach to it, and it will be read, and the correct transfer RNA attaches the amino acids, and this is where the protein chain is going to be formed. So let's go through these steps in much greater detail, starting with transcription. So there's 3 steps to transcription, and the names of the steps for transcription are the same as the names of the steps in translation. And the first step for both is called initiation. So in this first step, the DNA, which is in its double helix, needs to be unwound by an enzyme, and that enzyme is a DNA polymerase. And it's going to attach at this yellow area you can see in this picture, which is called the promoter site. It's like the start signal, okay? And so the DNA polymerase is looking for this start signal, and that's where it binds, okay? Once it has bound to that, what happens is we get a little bit of untwisting of the double helix, and we form a little bubble in there. And it is the strand of DNA in the bubble that are going to be transcribed. But you notice when you untwist, we have 2 strands. Only one strand is going to be the template and be transcribed. The other strand just kind of like hangs off like your friend on the side that's a third wheel on a date. Okay, not involved in the process whatsoever. So after initiation, we go to the second step, which is elongation. And so that same enzyme, that RNA polymerase, is going to continue traveling, and it's called RNA polymerase because we're making RNA. Okay? So it's going to travel down the exposed bases on the one strand of DNA. And it's going to be adding the complementary bases. So for instance, if there's a cytosine, it's going to bring a guanine. If there's a uracil, if there's an adenine, it's going to bring a uracil, because remember, DNA contains thymine, but not RNA. RNA uses uracil to bind to adenine. So it'll bring a uracil. And so it's going to bring all these complementary bases. And once the RNA base is next to its DNA base, then little hydrogen bonds are going to form to kind of hold them in place. And that's going to continue along that whole bubble as the RNA polymerase moves along. And so when you're working on this part, don't forget it's RNA bases that are attaching to the DNA template. Okay? And this is going to continue until it gets to the stop sequence. And in this picture, that is shown as this green area of the DNA. And so once it hits the stop sequence, we are in the third step, which is called termination. We are stopping, okay? And so what does this mean? Those weak hydrogen bonds between the RNA and the DNA, they're going to break so that the RNA strand is now separate from the DNA strand, totally detached from it. And so now we have the bubble with the two DNA strands, and what's going to happen is their hydrogen bonds between complementary base pairs, they're going to reform. And once that's done, then it is going to rewind as a double helix. So here is that here's that strand of RNA that was just formed that we are calling pre-RNA. And when you look at this part, you can see like there's blue parts and there's gold parts, okay? And that's because we have two different areas. We have an area what are called exons and an area of introns. So what are exons and introns? That's a great question. An intron is a segment of nucleic acid that's not going to do any coding. It's just inserted in there as placekeepers, just in case stuff goes wrong. But we have to get rid of it. It's extra stuff. The exons, those are the ones that we want to leave the nucleus. We want them to exit through the nuclear pores and be coded for. And so they're going to have to somehow attach to each other so that we can make sense of this mRNA molecule. So here's how this is going to happen. It's a process called splicing. So in RNA splicing, basically we are doing that editing job. If we look at the entire RNA that was just formed, what you'll notice on each end of it, there's kind of a cap, okay? Kind of like the little cap you have on your shoelaces, so that part can wear down and it's not gonna ruin the shoelace itself, you'll be able to tie it. So these little caps there are kind of protecting the code that is deeper in, so none of that accidentally gets lost. All right. Then what we're going to do is we are going to cut out the introns, the gold areas, the non-coding regions, and then we are going to splice together all the exons that were left behind. So we're just going to kick out all those little introns, and then you can see we have what we call messenger RNA. It's not the pre-form, but this is the mature form of messenger RNA. So it's still got the cap of the tail, and then we just have exons in between it. And this is what's going to exit through the nuclear pores. And so with that, we are completely done with the first process of transcription. Okay? So here is a review slide. I ask you to stop it, review this process, and make sure you use all of these terms when you're walking yourself through the process to make sure you really understand what's happening. Now, let's look for a second at that messenger RNA. So it has all these nucleotides, all these C, Gs, As, and Us. 3 of them in a row are called a codon because it's going to code for a specific amino acid. So it's the messenger RNA codons that are coding for the amino acid. And if we take the 4 base possibilities, A, U, C, and G, and we do like all the possible combinations, there's 64 possible combinations. But as we learned earlier, there's only 20 basic amino acids. So If each amino acid was only coded by one codon, we would only need 20 codons. So that means we have an extra 44. So what that means is a lot of amino acids can be coded by more than one codon. Not everyone, but a lot of them can. And some of them can be coded by 3, 4, or more. So when the cell is looking to start interpreting these codons to add amino acids, it's looking for a very specific codon, what's called a start codon, and that is an AUG. So when we're starting at the start, the cap and the tail part, and we have these codons, it's going to keep on reading until it finds an AUG. And at the AUG is where it's going to start, and AUG always codes for methionine. So here's our AUG start codon, and methionine is always going to be the first amino acid. So every single protein chain of all those 20 different proteins, they all start with a methionine. Okay. After that, it's going to continue going in the triplets. All these codons are being read, bringing in amino acids until you hit what is called a stop codon, a stop sign. Okay, so there are 3 codons that are stop signs. So when it hits the stop sign, there is no protein that is attached. So there's 3 stop signs. You don't have to have them memorized because they're in the table. So for instance, UAG is one of them. It's a stop codon. There is no amino acid. Now, students, they hit the stop codon, they realize there's no amino acid, but a lot of you then continue translating the rest of the messenger RNA. Once you hit the stop sign, you are done. There's no more work to do. So this particular amino acid chain in the pink example is only 3 amino acids long: methionine, lysine, and phenylalanine, period. Stop, the end, that's all there is. So you're done translating once you have hit one of the 3 stop codons. So here is one way where you can look at the table with the codons to figure out what amino acid. You start on the left side where it says the first base of the codon. So just pick one like CAG. Okay, and so you would go here to the yellow part, which is C, then you would go across to where the gold A is, and you would be in this square, and you're looking here for CAG, and what you notice is CAG will code for glutamine. Okay. So if there is a messenger RNA codon that's CAG, that means a glutamine is going to be attached to the amino acid chain. So I've put a little sample over here with 7 little codons, and because it's hard to read in the double helix, they are rewritten out over there on the side. So practice seeing how well you are at translating these from the messenger RNA to the protein that's going to be attached, okay? So stop this, go through them, and at the end I will give you the answers so you can double check with yourself, all right? All right, thank you for doing it, and here are the answers. And now let's talk about some transfer RNA, the 3rd type of RNA. Okay. So this is a single strand of nucleotide, but the hydrogen bondings between them make it look kind of like this clover with a stem. So 3-leaf clover with a stem. And what you'll notice that at the one end, we have the start and the end, and directly opposite from this set of bases doesn't bond with any other bases, so they are left exposed. And this is called the anticodon. Remember, codons are only on messenger RNA, so this anticodon is on transfer RNA. And so this anticodon is going to bind to a codon. So what do you think CUC will bind to? Well, C is complementary to G, and U is complementary to A. So this anticodon CUC is looking for a codon that is GAG. G. And so this is one binding site, but there is a second binding site at the other end, and this is where the amino acid will be attached. Now remember, the amino acid is coded for by the messenger RNA's codon, not transverse RNA anticodon. So you would not take this CUC and go back to the table to figure out what it would be coding. You would not be going and adding a leucine. You would be going GAG. So that would be a glutamic acid is what the amino acid would be. So we'd have a glutamic acid at this end, and the anticodon CUC would attach to a GAG. Okay, so this is what is doing the actual translating from the nucleic acid language to the protein language. So in order to do that, it has to interact with an anticodon, and the anticodon has to interact with the codon. And so this is showing you, remember I said methionine was always the first amino acid and the codon was at AUG. Well, that means the transfer RNA would have to be AUAC. So the U would attach to the A would attach to the U, the C would attach to the G, these lovely hydrogen bonds, and then we would be able to start with the methionine. All right. So the ribosome is where this bonding of codon to anticodon occurs. All right. And so this one shows the messenger RNA across the top, and now you can see the little transfer RNA that matches the sequence and does the complementary bonding, how that's ready to come in and attach to it. So let's look at the ribosome to see how this happens. So as you know, the ribosomal subunits were formed in the nucleus, and we had a big subunit and a small subunit. And they couldn't join together because if they joined together, they would be too big and wouldn't be able to lead through the nuclear pores. So they come out through the nuclear pores, and then the two are going to attach to each other. And the small subunit is actually on the bottom part, and the big subunit's on the big part. And the small subunit has a binding site for a messenger RNA molecule to attach. And the large subunit has three areas that bind, and they are called the A site, the P site, and the E site. And basically, they're going to be, binding has to happen in that order. The A has to be first, then the P is second, and then the E is third. Okay? And this is where our protein chain will grow. So let's see exactly how that happens, because it's a little complicated. So what's going to happen is the ribosome is going to move along the messenger RNA strand and until it finds that AUG, and then in the add side, we're going to have the transfer RNA with the methionine going on there. Gets added to the add site. And then for the next one to add, it has to move over to the P site because P is where the growing protein is. So the methionine is going to be the start of the growing protein. So here we see that first transfer RNA sitting in the middle, and there's nothing to the left and nothing to the right of it. So it's ready to be joined by the second transfer RNA. Here's one that's showing that two have already gone through, and here's a third amino acid going in. Sorry, a third transfer RNA coming into the A site. So the P site is where the growing chain is going to happen. Notice methionine is always the first amino acid. Now this transfer RNA is going to go into the A site. It's bringing a phenylalanine going on there. And then what's going to happen is this part of the protein chain is going to come off of the transfer RNA and attach to the new transfer RNA. And then once this one has lost its protein, it's going to shift over into that third E site. So think the A site is for adding a new transfer RNA. The P site is where the protein is, and the E is the exit site. So here you can see 4 transfer RNAs. This one has just left. Notice there's no more proteins attached to it. Its protein is at the bottom of the chain and the one that is in the E site. Here was the last one that was added in the A site. So this protein chain is going to detach. And you can see right here, this yellow, this is the bond between the bottom of this protein chain and the new amino acid that was there. And once it's found there, then it detaches from the transfer RNA. And here's an incoming one wants to go in. The A site is full, but as soon as this detaches, it's going to go to the E site. Then this one's going to go to the P site, and then the new one can go to the A site. All right, so let's talk about translation once again, step by step, to make sure you get it. First step, same name, initiation. All right, messenger RNA has to attach to the small subunit. The large subunit is going to attach with its A, P, and E sites, and the transfer RNA carrying methionine that is complementary to the AUG is going to attach to it. And because this is the actual start, this one actually doesn't have to start in the A, it actually starts in the P site because it already has the protein attached to it. After initiation, what step do we have next? Yep, that's right, elongation. So now here's where we left off up there. And now we have a new transfer RNA with a new amino acid that's complementary to the next codon. It is going to attach in the A site. Then we're going to have bonding between the lowest amino acid in the P site and the amino acid in the A site. And it will detach from the transfer RNA in the P site. And as you can see in this picture, it is attached to the one in the A site. That is the signal for the transfer RNA in the P site to move over to the E site and the one in the A site to move over to the P site. So as you can see, that one that moved over to the E site, that's like an ejection button. And you are gone. You are out of here. And then you notice that once the one in the A site has moved over to the P site, we have room for a new one to come in. And so this process continues repeatedly until we have the termination step. We get to one of those 3 stop signs. So this is showing this protein chain has 4 amino acids and it has hit a stop sign. So at the stop sign, no new transfer RNAs can attach because nothing codes for a stop sign. All right. And so what happens then is the amino acid chain is broken off of the transfer RNA. The transfer RNA moves to the E site, it gets ejected, and now we have a free floating in the cytosol amino acid chain. The ribosome splits back up into its small and large particles, and the messenger RNA breaks off the small subunits as well. So that is translation. So take a moment, stop it, talk yourself through the process, and then also answer these questions down at the bottom to see how well you did, how much you got. So when you're done, if you want to, here is an overall thing that pulls everything together. There's a bunch of core ideas I typed out for you, as well as some fairly basic questions for you to look at. So I want to thank you for all your hard work in this class, and I will see you when we meet again for our next topic. Bye-bye.